Here we go. I shall share my screen just a second. That one, share. All right, welcome everybody to the final session of the Plant Cell Identity webinar series. If I manage to change my slides, here we go. We set out a year ago to address the question, what is a plant cell type and what is a plant cell state? And to explore these concepts, we've had an amazing set of speakers throughout the last year to discuss their research and their ideas about how to use the terminology. Many of these talks are available as recordings on YouTube, the QR code and the link you can see here. We also, when we started the seminar series or webinar series, we started a Padlet to keep the community conversation going beyond individual webinars. And there we have some really cool conversations going and chats about how to use the terms, and also follow-up questions from the Q&A of each webinar is posted there to even further fuel the conversation. But please continue using this resource to keep discussing and keep your ideas going and share with the community. And today we have the panel discussion. We have three wonderful panelists. We have Olivia Wilkins from University of Manitoba. We have Bargmann from Virginia Tech, and we have Marc Libault from University of Missouri. And as moderators, there is me, Kaisa Kajala from Utrecht University, and Ben Cole from JGI. And how this is going to run today is, stop sharing, is that we shall, now I've lost the Zoom, sorry. Here we go. Sorry, I lost you briefly. The planned timings are that we're going to have first a brief introductory round from the all of our panel speakers. Then we are going to discuss the overarching questions of the webinar series. So we have basically tackled three main questions, which are: why is it important to define cell types and states? How are you defining them now? And how could you better define them in the future? And after that discussion, we have some more questions lined up. Or if you post your questions and conversation topics to the Q&A, we will start discussing the topics suggested by you. Ben, as my co-moderator, would you like to say something?
No, nothing in particular, but I'm really excited to see where this discussion goes. And yeah, I think it's a fascinating topic. And I wanted to thank also the panelists that came here, as well as all the other speakers that agreed to give their time throughout this series for some really insightful talks and some discussion.
All right, so let's start with the introductions from the speakers. So Olivia, would you like to go first?
Sure. Thanks so much. Yeah, no, I've enjoyed this series so much. I feel very excited to be a part of the discussion. So in my lab, what we're interested in really are development altering stressors. So these are stressors that don't lead to tissue damage, but that really lead to altered patterns of development with respect to how big the plants get and how much yield they produce. And the reason we're interested in these stressors, even though they're relatively mild, is because they're extremely widespread, and because I think they include a set of conditions for which genetic solutions are likely to have a really high impact. And so in particular, what we have been working on for the last few years are the effects of continuous water deficit and salinity on developing rice leaves. And so the treatments that we apply make the plants about 30% smaller, and they do about 20% less carbon assimilation. But in the period of our experiments, importantly, we don't see anything dying. There's no sort of damage to the tissue that we can see. So we've been working with really young leaves that have just emerged beyond the leaf sheath. And so our specimens include cells from the very base of the leaf, through the division zone, expansion zone, and maturation zone. And the methods that we've been using to sort of query this system that I think are relevant to today's discussion are single nucleus RNA sequencing. And we do this because we want to understand how stressors impact development, and to understand how different cell types and developmental states experience stress, because ultimately our aim is to guide crop improvement strategies using these data. So that sort of is the perspective that I come to this task.
Thank you. Then Bas, brief introduction from you.
Sure. Thank you very much. Yes, I'm Bas Bargmann. I'm an assistant professor at the School of Plant and Environmental Sciences at Virginia Tech. Very pleased to be here and asked to be a part of this panel. My interest in cell types dates back to 2006 when I started as a postdoc with Ken Birnbaum back in the day. Back then, I was looking at cell type specific responses to external or internal stimuli. Specifically, I was looking at auxin treatment and cell type specific transcriptomic responses to auxin treatment. Funnily enough, I did not work on regeneration at all while I was with Ken, but picked that up later on. And so in our lab currently, one of the things we're very much interested in is looking at regeneration specifically from protoplasts. Right, we're studying that both in the model system Arabidopsis as well as trying to get it going in different crop species. And so some of the guiding questions there are about, I guess, right, de-differentiation, redifferentiation, acquisition of new cell fates, and pluri and totipotency in plant development, where indeed having ways to define cell types can be crucial to determining the outcome of certain treatments. And so I think this is a very interesting discussion and something that I'm sure will be debated for a long time, but I think it's important to have this discussion with a great group of people like this.
Thank you. And then Marc.
Thank you very much, Kaisa. And thank you very much for your invitations to come to this. So I'm currently a professor in the Division of Plant Science and Technology at the University of Missouri, Columbia. And I've been interested about cell biology, really at the single cell type level since 2005, so almost like that, Bas, when we are working as a postdoc on shaving soybean roots to collect root hair cells to study the early stage of the infection of the plant by symbiotic bacteria involved in nitrogen fixation. And since those days, I've developed in different universities, first University of Oklahoma, a focus on the biology of this cell type, gaining more frustrations, about accessing accurate information from this unique cell type. And so I think where it was really a game changer was back in 2018 when we start to move towards the use of single cell biological applications and working with plant nuclei in this case to develop an understanding of both gene expression, but also chromatin accessibility. And now we are applying this type of technology extensively on different crop species, but still with a strong focus on legumes and their response to those nitrogen fixing bacteria. And so what I learned, I would say, during the last few years is I think context is important, and we have expanded our capabilities around whole plant single cell omics. So that gives us a very different perspective now on the programs that drive the biology of different cell types, and as a consequence on, thinking more about those definitions of cell type and cell state. So it's going to be very lucky and very exciting conversation this morning.
Okay, thank you for the introductions. So first question indeed is why is it important to define cell types and states? So we could just say that coming up with uniform definitions is too difficult. Let's just not even try. Why do we need these cell type and states definitions in place? Who would like to open?
Sure, I guess I'm happy to jump in first, and since that seems an easy way to go without other people stealing my ideas. No, just kidding. I think point one obviously is communication, right? We want to be able to communicate with each other and clearly state what we're talking about when we use a certain cell type name. And so I think, right, definitions are a crucial part of just communication and semantics, if you will, so that we can make sure we're on the same page and are talking about right, similar interpretations of what a cell that we're discussing entails. But secondly, I think it's very important for us to think about defining cell types, in that it'll also teach us about how plants themselves might define or determine what a cell type is, right? And as a developmental biologist, I guess that's one of the big questions I'm interested in, right? How do we figure out how a plant itself changes or endows certain cells with a fate? And how do we know that if you yourself are trying to change the cell's fate, for instance, how do you know you've achieved that if you don't have a measure of identity somehow?
Marc, Olivia.
Okay, I can continue. So when I think about cell type, I'm trying to, transfer the way we have been thinking for decades on plant organs. Everybody will acknowledge a root is not the same thing than a leaf. It's very obvious. And the function of those two organs is, of course, quite different. Now we have access to more resolutions. And so what we can, apply as questions on organ scales, you can do the same thing now on plant cells. Different cell types, different function, and they work together basically to fulfill overall a global organ, but also organism function. So I like to understand, the biology of those cell types because I think we can learn a lot about genetic programs and potentially apply this knowledge to make, I would say, more targeted, but maybe more impactful changes in the biology of key organisms, like, for instance, our crops. And so that's basically where we need to develop this foundational knowledge on plant cell type, potentially cell state, even if I think there's a lot of debate on these definitions, and we can speak about that today. But trying to get a better understanding of what makes a cell type unique, biologically speaking, and how we can use this knowledge to improve their biology.
Yeah, no, I agree really with everything that has been said. Although I'm going to add sort of a slightly different perspective, which is I think during the seminar series, there's been really, I think, so many different opinions and different sort of research projects and approaches that have really emphasized the need for sort of special cases, and that sort of if you're taking sort of a developmental approach or an evolutionary approach or a stress response sort of question, you maybe have different reasons for sort of setting some differences in terms of the criteria that you use for defining cell states. And I think there's absolutely zero risk of us ending up in a situation where every researcher is defining their cells in totally different ways. I don't think that that's a true risk. And so I think that when we start thinking about variation that might exist in the field, it's in a fairly limited context, and I think that there are going to be some things that are, obviously good practices and some things that are going to be obviously poor practices for this. But I think that realistically, we have to be, in the sort of, this series has been really far reaching, but in fact, it's only been 12 speakers or something, you know what I mean? And so the field is, there are lots of people working on all sorts of different questions. And we've seen really different opinions on some of these questions that have come up even amongst the 12 of us, or the 12 or 15 people who have given the talk during the series. So I think that we need to have definitions, and I think that we need to be explicit in our work when we're publishing and when we're talking about how we've defined things and sort of not only sort of technically how we've defined them, but why we have defined things the way we have, so that we know what we're talking about. But I think that this allows for some flexibility, but I don't think that we really are at risk of having, an infinite or as many definitions as there are researchers. I think that for the most part, there will be largely overlapping sort of definitions of things. So I don't know. I was interested to get all of the different perspectives we had during this series.
Thank you. So actually, I was rewatching some of the seminars. Don't want to say just last night, last minute prep, but some of them are very fresh in my mind. And Elliot Merowitz was mentioning that if we talk about cell types, maybe the way forward is to mention how it's defined. So cell type defined by transcriptome or proteome, or like give the sort of level of readout that you're using to define the cell types. So next question on our list is, how are you actually defining your cell types at the moment and cell states? So what goes into your definitions, and possibly even, can you give an example of a definition of your favorite cell type? Who would like to
go first this time?
Buzz again.
Oh, I'm happy to go again, but I was going to say I'm going to let somebody else go first this time.
No, go for it.
Okay, I will go for it. Yes, well, I think cell types can be defined on very many different levels, right? So we can look at the function and thereby group cells in types, or look at anatomy, right? So where in the organ or tissue is the cell spatially and in relation to other cells. You can think about the origin, right? What is the lineage of the cell? Where did it come from physically, but also maybe even on an evolutionary level, you can think about it on that level as well. But of course, what we've been doing more and more is just molecular profiling, right? So looking at epigenetic, transcriptomic, proteomic, metabolomic data sets, and using those as fingerprints, as you will. And I think that is right. Obviously, a trove of data that can be very precise, granular, and I guess the big question with such huge data sets becomes, where do you cut it off, right? How granular do you want it to be? You can make 1,000,001 different groupings and end up with an infinite number of cell types. Then, as far as cell state, right, which I think has been an interesting conversation in this PCA topic, is also an interesting question, right? What is cell state? So let's say two cells are based on some molecular profiling. Let's say transcriptomically, they're 90% identical. I don't know, I'm just picking a random number. But if you now give them a certain treatment, you might find out that actually, right, half of them react one way, half of them react another way. Does that mean that they were not the same cell type, or are those two different states and therefore responding differently to a similar stimulus? But yes, in the end, what we look at a lot in the context of my work is indeed this transcriptomic profiling, right? And apologies to the protein and metabolite people out there. But that is just such a relatively easily obtained data set for me anyway, that we base a lot of our definitions just based on which genes are being expressed, which marker genes, and orthologous marker gene groups perhaps as well, right? Looking between species.
All right. Olivia, would you like to go next?
Yeah, I think that, again, there's so many interesting ways of doing things. I think, though, like Baz mentioned, we also do a lot of molecular profiling of cells. And so this has sort of strengths and weaknesses, as does any method, in that, we're able to capture a large number of cells. We're able to sort of make a fairly good number of measurements about each of them. But we still encounter some situations where we can't actually transcriptionally define some cell types that we know exist anatomically. And even with, we have, in one case, more than 110,000 cells, and there are some cell types that we cannot distinguish. And so I don't think it's sort of a matter of coverage, and I don't think it's that we're failing to sort of actually measure those cells. I just think that they just really aren't that transcriptionally distinct from one another, that they vary in other attributes. And so I think that one of the things that we've really been thinking about is sort of how to either is it possible to sort of extract that information from the data using another method than one we've thought of already is one of the questions. Is it a matter of finding a really good marker set for sort of one of these subsets? And then, the other question, and I think this is probably the more likely answer, is like, what other measurement should we be making? It's not RNA based that would allow us to distinguish between these things. And I think that to me is going to be, like many other labs, we do our experiment, we define a cell type based on, or a cell state, or whatever attribute we're thinking of based on the transcriptome, and then we go on and do in situ hybridization to show that it actually that we're finding it where we thought we were. But I think that really it's becoming very clear that having sort of measurements from other sort of parts of plant response and other parts of sort of the plant biochemistry is, I think, really going to be critical for actually making some of these definitions a little bit more meaningful.
Yeah. Can I ask like a clarifying question? So you're describing a situation that you have like a clear anatomical or like a functional difference in between the cell types you have in mind, but then transcriptionally you're not able to tell them apart.
Yeah, exactly.
Yeah. So then your cell type definition still relies on the morphology of those cells.
Yeah, and so then the question is, what do you label them in your data set? It's sort of like this plus probably this, but we can't, and so you end up with sort of a higher level label than you might like, because it's not that I don't think we've got those cells in the data. We just don't have a meaningful way of annotating them or distinguishing them from one another yet. So, and I don't know that more transcriptome is going to help.
Can I ask another follow-up question? Like, how important do you think, I mean, if it's not a cell type that you're actively interested in, how important when you're collecting these data sets is it to try to, name and define everything that you've collected? Or do you think that it's okay to, just say like, I'm going to throw all this other data away and just focus on the things that, this study is interested in?
Yeah, well, I think you're like with these data sets, you're never going to describe everything, like just in that it would be a nonsense paper. What would you be really saying if you were to take that approach? So, yeah, I think that my reason for wanting to be able to at least label them is to make them useful to other people, because I think that one of the things that we have access to that not every lab in the world has access to is people who are really good at asking these sorts of questions right out of the gate, and so if we can provide at least some level of labeling to the samples when we are making them publicly available, I think it makes it a lot more useful for other people. And so that's where I think the value would be. But yeah, certainly in terms of, could we, could we publish the paper? Yeah. Is the paper submitted? No. So, I don't think we're sort of at the point of, the ease of publication, but I think that, reasonably, could we publish this without sort of annotating all of these things? Yes. And I think that probably it's not reasonable to label every cell, and that there are going to be truthfully a fairly large number that we can either label only at a very high level or that we just really don't know what they are. And I think leaving those unlabeled is a very reasonable approach. But yeah, no, I'm thinking more sort of to make it useful to others.
Thank you. Marc, how do you define cell types and states at the moment?
I like the fact that first we need to, as mentioned before, we need to really divide those two concepts. Things are not the same and they need to be handled in very independent ways. For cell types, or everything we have seen so far, they are mostly defined based on anatomical, physiological, and functional information that we gain for many, many years from researchers. And we use this information because we are comfortable with that. So it's really a good and easy way for us, to appreciate our cell types in the context of those anatomical and physiological information. That's why I believe, the interactions between single cell applications and spatial omics is going to be very important because it helps us to bridge those two types of informations, the new ones with single cell resolution molecular data, and basically what we learn from many years of observations of different organs and plant species. So that's really for the concept of cell type. Saying that, I think there's always interesting exceptions. I was digging recently in comparing the transcriptome of guard cells, but also hydroblast from the leaf. Amazingly, they are so similar, but their function is so different. And their location in the plant leaf is, of course, very drastically different as well. So I really think we need to combine those different informations together to better establish the definition of cell types. I think cell state is, I would say, even trickier than that. And maybe we can think from a philosophical point of view, why do we need to define cell state necessarily? It's very subjective. Everybody will have a definition. I think Olivia and Vas already mentioned that quite clearly. It's very subjective. It's really depend on the kind of information you will consider to develop your cell state definition. Right now, of course, with new technologies, we have, I would say, at least a chance to answer some of those questions. But maybe in 10 years, you are going to have, again, a new breakthrough, and maybe the definitions will evolve again. So I think it's going to be a constant change in the definition of cell state, and we need to be ready for that. We love to put our informations in bins and try to categorize things, and we need to be cautious about that. I think we need to give much more flexibility. What we need for the concept of family, genus, and species for living organisms, or even for our genes, we like to put them in transient factors, receptors. Okay, that's great. But then after, you're going to see all these kind of multiple functions and subdomains, and we try to always categorize things. Can we do the same thing with cell state? I think it's going to be complicated and very subjective. So we need to be cautious there and give a lot of flexibility in the way we are thinking those definitions of cell states.
All right. Just to give you panelists a little bit of things that I've heard discussed in the webinar series. So for the cell states, we have heard some talks to discuss these environmentally responsive cell states as per discussed by Marina Oliva, and how they see these two different lineages of the same cell type. So the containment of the environmentally responsive states and the non-environmentally responsive states. That was very clear in her data. We've heard there's a quite obvious consideration of the developmental states. So most easily in the roots, you have the meristematic elongation differentiation zones that are considered states. There are the cell states, so the cell cycle and those states. But I've also heard suggestions of the position. So for example, again, a root example, in endodermis, the radial position affects the transcriptome and positioning of the passage cells and that sort of thing. So there is also positional information that can affect us, affect how the profile is. So how do you guys feel about these being different states? There's also actually, before I pass it back to you, there's also somebody mentioned evolutionary states. So if you do a comparative work, there are sort of cell types that are not quite functional in a certain role, but are sort of accumulating or in the process of losing certain functions. So also evolutionary states. So how do you feel does that encompass what you think about the cell states? Marc, maybe back to you.
I can start this time. I fully agree with that. I think, cell-to-cell interaction, but also with the environments. And basically, we talk about the environment of the cell. It could be biotic, it could be abiotic environment, but it's going to have a massive influence on the status of your cells, how your cells behave. You can change one or another, it's going to have tremendous impact on the biology of the cell. I mean, we need to investigate that deeper, but when you consider the root system of a crab species, you're going to find multiple layers of cortical cells. Some of those will interact with the epidermal, some others will interact with the endoderm, some other will interact with other cortical cells. I cannot imagine they are all the same. They communicate, those cells communicate together. They are going to have some slightly different patterns. I think that's why it's going to be interesting. And again, right now, those patterns, we are limited to their transcriptome. Almost. I really want to go deeper into, the kind of informations we can gain. I think that, protein localizations and RNA localizations are going to be a very interesting also way to assess how different cells can influence their biology as well. So I think all those kind of interactions between cells and their environment is going to influence the concept of cell state. Now it's always, the question of where do you want to move from a definition of cell state to cell type. If you go back on this continuum of differentiation process along the route, at one point you are going to see very drastic differences between those dividing meristematic cells and the end product, which is a differentiated root cell. At one point, where do we put basically the line to say now we can speak about a different cell type and not anymore a different cell state compared to the neighboring cell? And that's why I think it's a very challenging definition here to bring. And that's why, again, I mentioned the fact that cell state is not an easy and very, very likely constantly changing notions that we will need to discuss over the years.
Yeah, I would agree with Marc that indeed it is. Well, I think it's currently still just a fuzzy line. Where is the difference between state and cell type? And right as we were discussing just now, right, the interaction with neighboring cells can be of great influence, right? And so in some cases it might be a minor difference, and in that case, are we talking about state? But another case might be a huge difference, right? So for instance, I think again going to the root, specifically the epidermis, right? I think most of us will agree that trichoblasts and atrichoblasts are different cell types. Well, they're both part of the epidermis, but right, the only or one of the defining differences there of that cell state is the interaction with the cortex layer, right? Are they overlapping a junction between two cortex cells or not? And that kind of defines whether you get root hairs or not. But then, yes, maybe in other similar contexts, we might not be able to tell such an obvious physical difference or transcriptomic difference for that matter. And indeed, where is that line? And I don't think I have a good answer, but I'm just pointing out that this is where we need to focus on. And well, I think as Marc was saying, also probably right, we'll have very different ideas 10 years from now as we do today. So it'll be interested to see where that goes and how we start pulling those two apart better than we currently can.
Olivia?
I could tell a story about leaves being different in terms of where they are and sort of how they respond, but I think I'd be aiming to make the same points that Baz and Marc have made already. So I don't think I have anything terribly new to add other than to just emphatically agree that it is a tricky definition. But again, I feel like, as Marc said in response to an earlier question, like, everybody can agree that a leaf and a root are different, like that's not sort of a difficult definition. And I think that even within the context of an organ, where you have the same cell types, that exist in different parts of the plant, that I think that there, to some extent, we would have some degree of comfort distinguishing between some cell states, for example, in the rice leaf, which is a system that we look at a lot. You can look at the transcriptomes of cells that are close to the base of the leaf and the transcriptomes of cells that are close to the tip of the leaf, even in a mature leaf. And they have very, very different, not only sort of underlying sort of when in the sort of control conditions, they have very different transcriptomes and very different photosynthetic rates, very different levels of sugars, very different, any sort of axis along which we can measure. They're different from one another, even at the level of cell types. But then when we apply a stressor to that, or really any type of stimulus, they respond really differently, and is it sort of that the response just looks different because the underlying sort of pattern of gene expression was different? That's certainly partly it, but we also see responses that appear to be, functionally different responses to the stressor because they experience the stress quite differently, based on the sort of the access to water, for example, or, how much photosynthesis they were doing at the time that the stressor was incident. So yeah, I think it's a tricky question, but again, I think that the answer is sort of in clarity of definition by sort of the person talking about it, rather than sort of a need for a sort of standard uniform definition, I think.
On the other hand, maybe some points we will be compiling a lot of the data together, and then at that point, maybe it would be good to start having this sort of annotation of, let's say, mesophil and added labels like leaf base, non-stressed, and you could have these sort of like states that are positional, environmental, developmental, sort of like the tagging system that we have in our electronic system. So you could have multiple tags added to like as the cell states to accompany the cell type as well. We have not yet gone to the evolutionary direction in the discussion a lot. So the very first talk in the webinar series was by Dennis Stevenson, and he showed beautiful examples from plant diversity. And one, I think that for a definition of a cell type, one thing he mentioned is that basically that to be a certain cell type, you come from certain shared evolutionary background, more or less inferring that same cell type doesn't evolve convergently multiple times. I think that's how I understood it. So how do you feel? Do you think that cell types can only evolve, like, does the evolutionary history of a cell type go into the definition of it? Maybe a tricky question if you don't work with a lot of comparative work.
I think, or if I may go first to answer this, because my answer will be brief. So I'll leave both of you lots of time to weigh in on this. But that's not sort of a perspective that we generally bring to the work. But again, I think that really from, we really, I think we're most often thinking about function. And so the origin, either developmentally or evolutionarily, is a very secondary sort of consideration in our definition relative to sort of what it is doing in the particular context. And so it feels, in seeing talks like his and like your own talk during the seminar series, I was like, oh man, we're missing out on this whole perspective that we don't bring into the work. But, and I was trying to think, how can we leverage, like sort of start incorporating these ideas into the work, but it's not something that we've made headway in. But yeah, and I'm interested to know if the other folks have done more with that than we have.
Well, we certainly don't necessarily take that into account very much or really work on that level. But as far as just answering the question, should that be taken along, I think it would be a clear yes. And I think there's a lot we can learn about what cell types have in common across species. And I think that will really help us narrow down the core identity. Again, not speaking from extensive experience on that, but just a thought I have, I guess.
I will bring maybe a different answer. When I think about evolution now, I'm thinking, of course, between species, but in terms also of function, evolution of functions. And when I foresee, for instance, I would take an example of, Bas, you mentioned the trichome. I will go back on this one. Yesterday, I was attending a very good talk where they mentioned this idea of search hyphae related to parasitisms of plants to plants. That was very interesting. Again, with this very unique tubular orientation growth. And then you can think about, the pollen tube. All of them will share basically a similar function of polar elongation. Can we imagine the conservation of a module, polar elongation module, that will be co-expressed in those three different cell types, leading to this polar elongation, highly conserved. So from an evolutionary perspective, it's there. But then you will have additional modules of genes that will confer the unique function of the pollen versus trichoblast versus hyphae. And that's create now a pattern of new function. So trying to have this kind of interaction of unique modules that could be shared between cell types, but the unique combination of the module will drive the biology of your cell type. And now how this will be conserved between species, I don't know yet. But that's the kind of concepts we are trying to think about and explore a little bit.
Cool. Thank you. And then the final overarching question of the webinar series, sorry for the detours that I took to poke you a little bit. How could we better define plant cell types and states in the future? So do you have any recommendations for the community how to do this better? Olivia, can I ask you for your opinion first?
Sure. Well, I think one thing that we have learned in sort of working through defining cell types and cell states in our own data is that using different types of approaches doesn't, in our experience, give radically different answers, but it does give different answers, like in terms of where sort of boundaries are set, when we're sort of identifying one cell type or another. And I think that we have found benefit in our definitions by looking at different strategies, and sort of the more expansive we have been in, and again, I'm talking about transcriptome-based sort of definitions of cell types, the more expansive we have been in terms of not just sort of using a limited number of marker genes, although certainly we use marker gene-based approaches for defining some of the cell types because that's the best that we've got. But I think that sort of considering more than a small number of diagnostic genes has been something that we've gotten a lot of benefit from. And partly because I think that for many things, they're like the diagnostic markers aren't necessarily functional or like that they may incidentally be high in the cell type, but they don't give you that much insight into sort of what is making that cell type what it is. So I think that being really looking at different approaches, I think incorporating them and sort of looking for where differences and commonalities exist has been really valuable for us. I think also, and one thing we haven't talked about, and I'm really interested to get everybody else's opinion, is there seem to be a lot of AI approaches for defining cell types, and that's not something that we have gone very much into, and I don't know if anybody else in this panel has. But I think, incorporating more validation assays, like that are not transcriptome based is something that, at minimum, sort of locating the cell types that you're defining using some sort of non-RNA based method, I think really adds a lot of value. But I think really in future, the thing that we're really going to benefit from is incorporation of sort of like high throughput, large scale assays that are sort of parallel to the ease with which we can measure RNA, but at the same sort of scale for measuring, for example, the metabolomes and the proteomes as well. I think that there would be a lot of value there.
Yeah.
And just the one thing I didn't mention either is like spatial transcriptomics, which Marc gave really good examples of how that would be incorporated. And I think that that's absolutely true and will become more valuable as the tools advance for that too.
Great. Marc, Bas, which one of you wants to go next?
After you, Marc.
Okay, I will go. So yeah, I think if we want to better define cell type first, I think we need to first come with a definition of a cell type and apply criteria to define them. So we talk about, the function of the cells, the molecular features, potentially the shape as well. You are going to find drastically different shapes. Each of those criteria could support the definition of a cell type. And then after, as mentioned by Olivia, identifying those molecular markers of a cell type identity is going to be maybe the most accurate way potentially to define a cell type. Again, I like to find, these kind of integrations of different approaches as a way to hopefully have a better picture of the diversity of cell types composing the upland species. Regarding cell state, I will try to be very cautious there again, thinking it's a very subjective topic of conversation. Everybody, I think, will have the right to put the line whenever they want to define a cell state, of course, with the conditions to clearly define the criteria. And that should be, I would say, a topic of conversation. And I think, those conversations will lead us to a better understanding of plant cell biology, which is ultimately the goal of the work we are doing. So I really don't mind to have again, those debates on what is a plant cell is going to help us to think and rethink over the years about those definitions.
Yeah.
Yeah, I wholeheartedly agree with both other panelists. and what they've said so far. And you're right, I think many of us are coming to this problem from the same perspective in that, right, we're using like single cell, single nucleus dissociative approaches where we're now trying to map these back to where they were originally, right? And I think that's where these definitions are very useful. Not the only reason we need definitions for cell types, but I think that's what drives a lot of us. And as mentioned, yeah, I think, right, being able to link those molecular profiles, let's say, back to some ground truth, either with right spatial transcriptomics, or perhaps even like including in your whatever organ that you're sorting or dissociating your cells or nuclei from, perhaps like a marker gene as well, where it's like, oh, I can look back, I know that this particular cell file has GFP in it. How does that match up with the transcriptomics or whatever you're looking at? I think also in essence, what we need and what we're working towards, or many of us anyway, is just a better understanding of cell fate acquisition. How is that determined? Having a better idea of how master regulators work, or the interplay between them, or modules, as was mentioned before as well. So I think as we progress in having a better understanding of cell fate acquisition and perturbation and retention, or what have you, change, will help us to make better definitions of cell types.
All right. So I don't see any questions from the Q&A yet, so I keep on asking questions that I come up with, which is fun, I find, but this is also, dear audience, your opportunity to get your questions in or conversation topics in. But in the meanwhile, I'm going to ask about the fact that depending on the cell type, basically the working definitions can be hugely different. So one of the examples that I find really wild is that the bundle sheet cells. So bundle sheet cells, as far as I know, are defined as the cells surrounding the vascular bundle in the leaves of angiosperms, I want to say. I don't know how far back it goes, actually. Here my knowledge fails. But it doesn't matter the function. So you have the C3 bundle sheet cells that are quite streamlined, and then you have the C4 bundle sheet cells that are huge, full of chloroplasts, carry out the photosynthesis, actually the Calvin cycle of the photosynthesis. And you have like huge functional and morphological diversity within the cell type, but they're all bundle sheets, and basically it is defined by the location, not the anatomy or the function. On the other hand, we have cells that are very much defined by their function and their morphology, and their location doesn't matter at all. So do you think that we should be building towards like definitions for each individual cell type? So like there's a definition for bundle sheath, there's a definition for endodermis, there's a definition for trichome, or should we have an idea of like what are the characteristics that we should try to outline for every single cell type that we define?
I can start. I think we need to establish some kind of, maybe not ontology terms, but a process for defining a cell type.
Actually, I'm going to step in and say that there are the plant ontology terms that are actually a really nice resource, but I think that they're getting a little outdated with all the knowledge that we're building from single cell sequencing.
The problem with not providing such kind of guidance in naming a cell types could lead to what we are facing to some extent with gene function. Sometimes they can be associated with the biochemical function of the gene, sometimes with a biological function. And let's say if you find in your, draw stress plant genes, genes related to biotic stress response, you raise questions and say, what's going on? But maybe it's just a mislabeling of the function of the gene that was first discovered as abiotic stress response gene that lead to this kind of, propagations of maybe misidentity. We want to avoid this kind of misidentity of plant cells. And having, again, some kind of more consistent way to name a plant cell type will be a gain for the scientific community. To some extent, I think the marker genes that Olivia mentioned before, supporting, of course, the conservation of plant cell identity between cell types, between plant organs, and extensively between plant species, that could be a commonality to drive the naming of plant cell types. Again, you will find exceptions, and that's something that needs to be established on the long term. So I think evolutionary biologists will have, I would say, a strong input in the way we want to name our cell types.
Bas, what do you think?
Yes. Well, I think this is a field that's obviously evolving and changing. And so I think we shouldn't be too rigid to try to lock stuff down before we fully understand things, or will we ever fully understand things is another question perhaps. But I think we have to be flexible and be willing to change. But an important aspect of that is that as we are defining our cell types in our work right now, that we clearly indeed indicate how we are defining that in whatever statement we're trying to make. So I'm talking about this cell type, and I defined it as such, so that 10 years from now, when somebody's looking at your work, they'll be able to put that in the context of the time and how you were defining it at the time, because I think it's bound to change.
Olivia?
Yeah, no, I think, these are all really insightful comments. I have a question though for Marc, based on something you said, like, in that my concern with some of the marker genes is that, that they sort of are not, that they are highly abundant in a particular cell type, but aren't necessarily functionally that important to their definition. And so I guess if we were to use something like that is as sort of the defining feature of a cell, what would we even call it? You know what I mean? In the sense that, like, if it sort of, what would be our sort of, would we start from a cell type that is well characterized in other ways that we have defined, or that also has this sort of diagnostic marker gene, and then propagate that cell definition label to other things that also have that distinguishing feature? Or would it need to sort of be propagated only in cases where, for example, an anatomical or functional feature was also sort of carried with it? Like, how would you envision that?
I think it will really depend on the nature of the cell types we are considering. I mean, based on the work we have done between organs, between species, those kind of trichoblast, guard cells, phloem, xylem cells, very well differentiated cell types with very unique function. Usually we don't have so many problems to identify them based on gene ontology and the activity of those marker genes. Whenever we start to move on plant cell types, maybe I will not say maybe less differentiated, but at least maybe a differentiation process we cannot capture as well. From a biological function perspective, it starts to become more complicated to find those kind of commonalities between organs and between species for the same cell type. So I don't have really a good answer, Olivia. It's just, I think we are going to learn from our mistakes. We are going to make mistakes, and that's totally fine. But we are going to refine those definitions over the years, and I think that's what we need. Again, we are scientists. We are looking for the truth, and we are still searching right now.
Yeah, I mean, just to chime in here, I kind of think as like the nomenclature and cell type definitions, that they're not so much definitions, but more like a model. And that model is useful in some ways, but most definitely wrong in a lot of ways.
Okay. I do have one more question from my side, is that we discuss about how to, like, what we should be aiming to do, but do you guys have seen like things that we definitely should not be doing? So instead of trying to think of like what is the correct way forwards, like, do you have examples that that we like mistakes that we really shouldn't repeat when it comes to naming cell types or talking about your UMAP clusters or these types of topics?
I think so some of these are anecdotal and some of them are things that I've sort of like experienced as a reviewer or a person whose work is being reviewed, where there's sort of overemphasis on a single answer being right, like in terms of how we approach it, and a need to annotate everything and to sort of define everything when there isn't actually good data for doing so. I think that those I think are mistakes. I think another mistake that is not using sort of the wealth of data that exists to define your things, and sort of taking sort of the first answer, best answer sort of approach. And it's just, I don't think it adds very much value to the field when people have taken that approach. And certainly it could be that, like, are you likely to change something from being an epidermal cell to, like being like, actually, no, it's not an epidermal cell? Like, probably not, like probably you're going to have a fairly confident definition of some of these things. But I think that really, I feel like the insufficient explanation of how or what approach was used and why is one side, and on the other side, sort of requiring that sort of a personally defined rather than sort of a community defined standard of how we expect people to define cells. I think that those are mistakes that I see happening right now, and yeah, I think that really we aren't at a moment yet, although I hope we are at one soon, where we can have sort of fairly clear definitions about, these are sort of the considerations for defining a cell type. You may use, this set, or this set, or if you're going to add another set of defining criteria, you really need to be well justified in doing so. But I think that we aren't there yet. And so I think that there's value in any use of the single cell data, for example, to be explicit in what you've done and why, and to be flexible in your reviews. Obviously, it's not an anything goes situation. Some answers are going to be wrong, but I think that there are still possibly a fair number of right answers that, as far as the field goes. So,
thank you. Marc, Bas, do you have examples, thoughts?
Not an example per se, but a thought is definitely that I think we can easily forget that we're just looking at snapshots, right? Well, I guess an example I would bring up is indeed, right, that I'm sure some of you will be familiar with the work that like Rachel Sheehan and others have done coming out of the Benfi lab, where they did, right, looking at the root cell types over developmental age, right? So seedlings of different age, and showing remarkable changes there. And so that's something we should keep in mind, I think, that looking at the same whatever organ a few days before or earlier or later may have tremendous impact on how we would now define cell types based on just whatever omics you're looking at. And so that again is why I would emphasize indeed this flexibility, need for flexibility, and not being too strict in your definitions.
I can add very quickly one more comment. I think it's one mistake will be to try to force to annotate all the clusters. I think it's totally fine if we do not know and be able at this point to annotate all the clusters. I don't want to create basically a misunderstanding or a misinterpretation. I think it's better to keep the conversation open rather than forcing the annotation of clusters. And so we need to, of course, communicate that very well with our peers, that we have a unique information we can provide, but we are still not in the capability to fully annotate and understand all the different cell types. And I think what Bas mentioned about the snapshot, I think is very true. We need to acknowledge it's just a snapshot, and we're going to miss very likely important information and potentially cell types still.
I think this is an excellent note to end this conversation on. Really good summary of a lot of the things that we've discussed today, and where what we all, I think, can stand behind. Not, as Olivia also mentioned, not, requiring completeness of annotations or making wild guesses when the data doesn't justify it. So just basically working with a bunch of our data. So I would like to thank all of the 3 panelists. I would like to thank Ben for co-moderating with me, and I would love to thank Natanella and Trevor for being the main engines behind this webinar series, and all the, of course, Plant Cell Atlas for enabling all of this, and especially Johanna Murray behind the scenes for making things happen. So thank you, everyone. We will post this video on YouTube quite soon, and if you enjoyed it, please share with your networks. I hope that the discussion would be useful for a wider audience than only those who were here today following it live. Thank you all.
Thank you, everyone.